Okay, yesterday we ran 3 proteome comparison jobs, one using a whole genome as a reference, one using FASTA files from genes that we were interested in, which are these genes, which are the lipopolysaccharide genes from Brucella, and the third one was a feature file, which were the proteins, the genes from this paper, just to show you the differences and what it looks like. So I know you're excited to see that job. I'm excited too. Let's go look at it. So there are several ways you can view a job in Patrick after you've run it. You've seen me many times click on the job monitor, but let's go up into workspaces, click down here, and click on My Jobs. I can also navigate to it if I go into my home workspace, but this just gets me there quicker. So I click on My Jobs, and these are the 3 jobs. So let's see what each of them looks like. To be able to see something, you need to highlight the row. And when you click on that, this vertical green bar gives you a couple of downstream actions that you can do. If you have a problem with the job, you can report an issue with it. If you want to view it, you click on the view icon, which is what I'm going to do right now. Okay, this is going to give me all the information I want about this particular job. I hope it's all the information you want. It's a lot of information. Up here are hyperlinks to some of the folders that are the files that we have here, and the ability to download different things from the job. Across the top is a breadcrumb. This is the name of my job. It tells me it's in this folder in my home directory of my private workspace. This is the job ID. This job started on this date at this time. It ended at this date on this time, and it was fast. So 6 minutes, 51 seconds. One of the things I like about the proteome comparison jobs is they turn out results pretty quickly, unless there's a long queue. Patrick operates on first come, first served basis. So even I do not get a special place in the queue. I have to wait along with everybody. Parameters. If you click down here, this will show you everything that you selected when you submitted the job. Okay, so we have a number of files here that I want to describe to you. Let's start at the bottom with this reference genome text. I highlight that, and let's click the view icon. And this just shows me what the genes in the reference genome that I used, who they were and where they are in that genome. This tells me that this is the accession number for the contig, and this gene started on that contig at that location, it ended at that location, and this is the particular gene. So it should have, there are like 3,000 some genes. It should have one for each of those here. Let me click and go back here. We also have that data for comparison genomes. So it's tied in here, and let's look at it here. And this is telling me against that reference, these are the genes that had the hits and how strong their hit was. So this is the accession number, the start and stop location. This is the percent identity of this particular gene in comparison to that one. Okay, so I'm sure you're thinking right away, well, that's so very helpful to have the reference genes in one file and the comparison genes in another file, and how am I supposed to see anything? Doesn't sound helpful at all. And normally I would agree with you, but don't worry, we have a solution and you're going to love it. First, I want to show you this diagram, which is the Circos final HTML. Let's view that. Okay, What I'm soon about to show you is the details behind this visualization. But this tells you everything you would want to visually recognize about the proteome comparison job. Up on the top is the color key, which distinguishes between bidirectional and unidirectional best hits. The bidirectional are more vibrant, and the unidirectional are less vibrant, and it shows you the tracks from outside to inside. This is our reference genome on this track, which it blasts against itself in this job, so it should have 100% similarity. Next, we have the Brucella melitensis Reb1 strain, and you can see that there are really strong hits here, but some are lesser strength. Then we have Brucella canis, an isolate from dogs. Now notice that there are some blank spots here, and when you see genes here, it's an indication, it might be an indication that this genome is missing those genes. However, when we submitted this job, we said we wanted to have 70% query coverage, 70% sequence identity, and an E-value of -70. So we're setting the bar kind of high for these genes. So there might be genes in that genome that would blast against it if we weren't being so picky. But that's the way I played that game, and these are the results. But it also indicates that you need to, when you publish it, you need to describe exactly what you did. And also you need to verify things. So you need to go looking to see if those things were actually missing. Next is the ovis genome, which also has a big blank. Now you might ask, well, look here, there's a blank here on Brucella melitensis. Does that mean it's missing a gene? No, what it's trying to do is reflect the position and the orders of the genes on the chromosome. And when you see it in the reference genome, like right here, that it's missing something, it's an indication that there's just an intergenic region there where no genes are called. The next one is the FASTA file for those LPS genes, and then the feature group. And as I've said earlier, these genes and these files came from this genome, so the hits should be strong. Now you may be wondering what is going on here and here with this break here. It starts at 0.1 and then it starts at 0.1 again. What's going on? Brucella melitensis 16M is a closed, complete genome. And when something is closed and complete, it means that the number of chromosomes and plasmids that an organism have, we have a contig that represents that, and that within the contig, each nucleotide is an A, T, C, or G, and there are no ends called for that, where it means the sequencer couldn't distinguish and determine what it was. The way this is visualized is this big part that takes up like 2/3 of the circle is chromosome 1, the large circular chromosome in Brucella. And the smaller one is chromosome 2, the smaller circular. Some people have told me when they have multi-chromosomal genomes, I don't want it to look like this. I want to see the individual chromosomes. The way I would do that is submit each of those chromosomes individually to the annotation service and then run proteome comparison on that. Okay, so that's what that looks like. Let's go back to the jobs folder. So we just looked at the HTML. This SVG is a scalable vector graphic publication quality that you could submit with a paper if you wanted to publish on this. And please cite Patrick if you do. I look at every paper that cites Patrick. I also tweet and Facebook about it, so it gives you some incentive. I'm trying to help you out in getting your paper recognized as well. But I see a lot of these in papers that are published that cite Patrick. There are a couple of other text documents in here. The karyotype, the large tiles, the legend, that's just, well, we can quickly look at that. The karyotype is just talking about the 2 large chromosomes in Brucella melitensis. The large tiles, another boring document that's, I don't think it's very useful. The legend, that one's good, that one shows you the color thing. But the most important one to me is you have the text and the Excel file of the genome comparison. This genome comparison file is taking the reference and each of the comparisons, and it's combining it in the most awesome Excel file perhaps that the world has ever seen. So let's look at it. I highlight the row, and this one I have to download. So I click download. It says, should I open this with Excel? You bet, because I'm a biologist. I love Excel. So open it, and for some reason, Excel is dumping it down there. And while it starts to resolve, I'm going to open the whole thing. Come here, you. Don't be shy. Don't be shy, little Excel file. Be proud in your beauty that we are going to show these people what you look like, because it is amazing. Okay, this is all the data combined together, and you notice it's keeping the colors too. So if I had that legend up, I'd be able to distinguish between best and bidirectional and unidirectional hits and all of that stuff. Let's go into view and let's try to make this a little bit bigger for everybody to see. Okay, it starts with the reference genome, okay? So all of this from here all the way to here, let me just give you a color highlight here so you can see that. That's our reference. When I go from here all the way to here, that's, let's choose this color. That's the first comparison genome. Here, all the way to here, that's the second comparison genome. So this is each of the column heads underneath that belong to that particular genome. So let's step through it, and I'll blow them up so you can see each one. This is the reference genome contig. So this is saying this is the accession number where the first gene on the reference genome is located. This is the size of that gene. Okay, if you look up here, it says reference genome amino acid length. This one is 341 amino acids long. This is the Patrick gene ID. Now be careful clicking on this because it's a hyperlink and it's going to take you back to Patrick and show you that gene. If that gene has an accession number in RefSeq or GenBank, we include it here. If in RefSeq or GenBank it's called by a different gene name, like Hemi, we include it here. This gives you the local Patrick family ID number. This is the global Patrick family ID number. This is what the gene is called in Patrick. Ugh, it's long. Our names are sometimes exceedingly long. So this is the start on this chromosome. This is the end on this chromosome, and this is the orientation. So it means it's on the forward strand. And so this goes for every single gene in this genome. And you'll notice like this one has a Patrick ID, but not a RefSeq gene, meaning it's called via the Patrick Annotation Service, but PROCA, which is used by GenBank, didn't call this gene. Okay, now we get to the more exciting part. Well, they're all pretty exciting. So this tells me, this is saying what type of hit is it? It's bidirectional. This is the accession number on this Rev1 contig, the accession number, that is the best hit to this gene. This is where it starts. This is, oh, I'm sorry. This is how big it is. This is how big the amino acid is. This is the Patrick identifier. If it had a RefSeq locus tag, it would be included here, and a RefSeq gene name, it would be included here. This is what it's called in Patrick. And then here's where things get very powerful. The percent identity. So it's got 100% identity. And you can see a couple of them are smaller. And this is the sequence coverage. So it's showing you who the hits are and how good those hits are. And here, let's start with Canis. When we get here, like here in Ovis 2, these guys didn't have any hits of that caliber. Remember that we set a high bar of 70% sequence identity, 70% query coverage, and an e-value of -70. So these guys didn't have any genes that met that bar. And this guy is missing a few too. And so you can already see that this could be a powerful way that you can look very quickly and see how close the genes in two genomes are, but also see large blocks of things. Because remember, this is walking down a chromosome, large blocks of things that appear to be missing. And you could look across the other things as well. Now, oh, look, when we get here to these LPS genes, remember we were looking at a specific FASTA file. So that's very limited. So you can see suddenly you can sort of get a feeling for why you might want to use this as a reference, because if these are the genes you're interested in, that's all you care about. And there's all this other stuff that you don't even want to have to wade through. So let's go in and look at what that looks like. So I'm going to go back to here. And actually, I want to go to my jobs page, because we've discussed everything I think we need on this one. This is the one with the genome. And you can get quickly to the beautiful colors and the Circos diagram by clicking here. Okay, and that's going to show us this. But then I also wanted to look at the other jobs so we could go back and forth between the files. So this is when I used a whole genome as a reference. Here, I'm going to go into my jobs. Here's the FASTA file. Let's view that. And then click the view icon up here in the upper right. When you get more specific on what you're asking for, the diagram changes, okay? And then this, you might think, well, it's going to look exactly the same as the last one, right? Let's see if from the feature group file. So this is the feature group. The other one is the FASTA file itself. This is the feature group. So let's view that. And then click the view icon. Okay. So let's describe what the differences are. Here's the feature group, and that was like 72 genes. What this is showing from outside, the first one is the feature group. Then we have Brucella canis, Brucella ovis, Brucella melitensis, Brucella abortus 2308, then the FASTA file, then the Rev1 genome, and then the genes from 16M, okay? So this is just showing where, because I took this feature group from Patrick, it's translating this into, it's trying hard to resolve it on the genome. So it's showing you exactly where all of these genes are in the genome. And you can see that there's a large number of LPS genes here. And in fact, one thing I didn't show you on the other diagrams is that some of these can be used as hyperlinks. There we go. And it'll open up and show you a particular gene if you're interested in it from that. But I'm not even interested in waiting for it to load because, so you've got this, which is showing you the things that you were interested in the context of this whole thing. So it's like blowing up these two lower circles. But I'm just really impressed with this, which is giving you all the data. This is a large gene here, and you can walk through it. Well, this is the FASTA file against itself, Canis, Ovis, abortus 2308, Rev1, and then the 216M genes there, or the FASTA file or the gene file. And that's how I really like the way this looks. And also, if we download, let's go back to the folder for it. If we were to download the genome comparison for this, so I say, yes, I want to open that. And here it is down here. So let's give it a chance to open because it's heavy because it's got all the coloring in it too. So we'll stretch it out. Come here. So then it's just showing you for those genes of interest, all the BLAST data. And it's amazing. I just love this thing. And I hope that you do as well, because it's an amazing tool and it's a great way to really dig into the data. So in the last video of the series, we'll talk about citing Patrick and what to do when you have problems and need to find help. So thanks for joining me and see you next time.